To treat SARS-related disease in humans, can we prevent protein priming and block that process to reduce, if not to totally prevent, viral spread and pathogenesis? SARS-1 spike protein can interact with cysteine protease CATBL or serine protease TMPRSS2 so that it can get primed and cleaved, enabling viral entry into host cells through membrane fusion. VSV was used in this study, and VSV was pseudotyped to express SARS-1 S protein or SARS-2 S protein. Vero cells were also used in this study, and these Vero cells do not express the serine protease, but only express the cysteine protease CATBL, as well as ACE2 receptor. E64D is a cysteine protease inhibitor, which works against CATBL. We see that with both SARS-1 and SARS-2, treatment with the cysteine protease inhibitor significantly reduced pseudotype entry into host cells and almost abrogated it completely. On the other hand, treatment with Camostat, which is a serine protease inhibitor, had virtually no effect on pseudotype entry into host cells. This makes sense because there is no serine protease active in this situation, and so treatment with a serine inhibitor should have no effect on pseudotype entry. In addition, adding the Camostat drug, the serine inhibitor, on top of the cysteine inhibitor did not further reduce pseudotype entry. This demonstrates that both SARS-1 and SARS-2 spike protein can in fact be primed by CATBL, the cysteine protein, especially in the absence of TMPRSS2, the serine protease. In this next experiment, Vero cells were transfected to express both the cysteine and serine protease. Interestingly, what we see is that treatment with just one inhibitor does not really reduce pseudotype entry as dramatically as it did before. On the other hand, you really need both drugs in order to really limit pseudotype entry. And so this means that the virus can use a serine protease to prime its S protein, but also a cysteine protease. And to totally block viral entry, in this situation, you needed both drugs, and this holds true for both SARS-S1 and SARS-S2.
Vero cells without the serine protease and with the serine protease which are generated are treated with E64D or Camostat mesylate 2 hours before infection with SARS-CoV-2. E64D blocks the activity of the cysteine protease, and Camostat mesylate blocks the activity of the serine protease. We are using these drugs because they are clinically approved for human use and because the cysteine protease and serine proteases are both involved in SARS-1 spike protein priming and cleaving the S protein into S1 and S2 subunits. Although both proteases can be involved, both are not necessary for SARS-1 entry. The question is whether SARS-2 requires both proteases or just one of them, and if so, which one?
At this point, we're all thinking, okay, these statements seem good, but they all also seem very conditional. You're using pseudotype virus, you're forcing expression of receptors and proteases in cells that normally don't express these things. So what happens when you actually use the wild type virus with a human lung cell line, which is one of the main cell types that are infected by this virus, and this cell type naturally expresses ACE2, the receptor that the virus uses. In the interest of time, I'm not going to show data where the authors compared Camostat, the serine protease, against E46D, the cysteine protease. In general, they found that Camostat was more effective than the cysteine protease. They also referenced data from other authors that suggest the same thing. And so now, Camostat became the focus of the study. I will list an example of one of these references below, where a clinical trial is currently being done using Camostat in humans at a university in Denmark. These ideas, with a focus on Camostat, are supported by mouse model studies for SARS-CoV-1, in which Camostat treatment reduced mouse mortality by about 65%, bringing 100% mortality down to 35%. So, in this experiment, human lung cancer cells that naturally express ACE2, the serine, and cysteine protease were first treated with Camostat, and then washed, and then inoculated with SARS-CoV-2 isolated from a human patient sample. So this is wild-type SARS coronavirus 2. Interestingly, what we see is that after 24 hours, there is a significant reduction in viral entry in the Camostat treated group compared against the control. Now, this is really exciting because this was done with wild-type SARS coronavirus, not a pseudotype, and in a human cell line that naturally expresses the receptors and proteases we are most interested in. And so this is really exciting because it suggests that Camostat could be highly effective in reducing viral entry and preventing viral pathogenesis in human patients, and that this could be a promising treatment option. One of the questions that is left sort of unaddressed is, why can't we do this in a mouse model? Well, unfortunately, there are no mouse models out for SARS-CoV-2 entry that also have humanized ACE2, and they are in development but not yet ready. So that's also a current barrier for properly studying SARS-CoV-2 treatment, especially in vivo. And so this is why we have to often reference SARS-CoV-1 or use in vitro data.
Calu-3 cells are pre-incubated with Camostat mesylate or are mock-treated, which are the control, and are then infected with SARS-CoV-2. Cells are washed, RNA is isolated, and genome equivalents are determined by qRT-PCR, which amplifies viral RNA. In qRT-PCR, expression of virus-based RNA is compared against a basally active housekeeping gene to determine the magnitude of viral infection compared against baseline expression of a constitutively active housekeeping gene.